ahead and get started on our next Ivan workshop on advanced MRI, MRS in brain and liver. At this point, I'll turn it over to David Rice to get us started.
Okay, thanks, Eric. And as Eric said, our leader today is Dr. Aaryani Tipirneni-Sajja from the University of Memphis, who is going to talk about MRS and MRI. I will hand things right over to Dr. Aaryani Tipirneni-Sajja.
Thank you, David. So this panel, hello everyone. This panel consists of 3 speakers. Myself, I'll be the panel leader. And we have Dr. Puneet Bagga from St. Jude Children's Research Hospital and Dr. Esin Isik from Bogaziçi University. And we'll be presenting on some advanced in vivo MRS and MRI techniques for brain and liver applications. So the format would be, I'll introduce each speaker before they talk, and please post the questions during the presentations, or you can ask them during the panel discussion. We will first finish the presentations and then discuss the questions during panel discussion so that the presentations are all done. And with that, I will introduce the first speaker, Dr. Puneet Bagga. Dr. Bagga is an assistant member in the Department of Diagnostic Imaging at St. Jude Children's Research Hospital. He also serves as the director of the Metabolic Imaging Research Hub there in the Diagnostic Imaging Department. His research focuses on advancing the field of metabolic imaging by developing clinically translatable non-invasive MRI techniques. And in this presentation, Dr. Bagga will discuss the development of a novel approach, quantitative exchange-labeled turnover MRS, for mapping cellular metabolism in vivo using proton MRS in combination with deuterium-labeled substrates. With that, I will hand over to Dr. Bagga to start off.
Thank you very much, Aaryani, for this kind introduction. And I must say I'm really privileged and honored to be a part of this, this meeting this time. And so thank you for this invitation. Well, I'll get started. I will be talking about MR spectroscopy because that's what I've been doing for past however many years. And as I was saying, I'm really honored to be here presenting some of our recent work, but it all stems from whatever we have been doing, whatever this community has been doing for over past 30 years or so. So I'll just introduce a little bit more about myself. When I was a graduate student, I was really interested in metabolism, but I did not know what to do. But I knew I was interested in NMR side of things. And also I wanted to understand more about brain. So I picked a problem, which is basically understanding the cerebral energetics in Parkinson's disease. So now we know much more about Parkinson's, but I'll just briefly take a few seconds to explain. It basically originates in substantia nigra pars compacta. It's a very small region enriched with the dopaminergic neurons, which have innervations into the basal ganglia, midbrain regions that have excitatory as well as inhibitory, connections with the downstream regions, namely thalamus, globus pallidus internus, externus, as well as the subthalamic nucleus. And they then have the excitatory innervations into the motor cortices. That's how the nigrostriatal pathway kind of dopamine kind of works to help us regulate the motion or make us do things. It seems like when you lose more than 90% of these neurons in the substantia nigra, all of this system goes haywire, and basically you lose innervations, dopaminergic innervations, and that excitatory inhibitory neurotransmission it's reflected upon that system, so to speak, in the brain. So not much was known about the cerebral energetics other than certain FDG glucose studies in Parkinson's disease brains before I was getting on this field. And so when we did this in mice, we did it in a very rudimentary way. Back then, we infused 13C labeled glucose in the mice, took for like a certain amount of time, 10 minutes to 2 hours, took their brains, extracted their metabolites, and then instead of running an LC-MS, which is the favorite approach right now, we did 13C MR spectroscopy to find out the enrichment in different metabolites, namely glutamatergic neuron metabolites, GABAergic neuron metabolites, and so on and so forth. And we found that indeed loss of dopaminergic neurons has a widespread effect in numerous other regions, not just midbrain, so to speak. So that's what led me on to the metabolic imaging sort of pathway. And since then, I've always been interested in developing newer sort of techniques for metabolic imaging, and MR spectroscopy is my favorite tool, so to speak. So as I was saying about 13C NMR, there's neater way of doing 13C NMR or MR spectroscopy. It's basically the same way you inject the glucose and you acquire a 13C MR spectrum, either an FID or a 3D readout, so to speak. And you basically monitor the turnover of metabolites, glutamate, of the label of glucose going into the different metabolites. And you can basically form a turnover curve. And then once you fit this data into your metabolic model, it gives you rates of, or kinetic rates of different pathways, namely neurotransmission, glutamatergic neuroenergetics, TCA cycling, and so on and so forth. So it's a very neat way of doing spectroscopy. But the only problem is 13C is, as we all know, 13C is very insensitive sort of nuclei. So and all our rates depend on the initial turnover, which is very, very noisy. So it lacks that kick for translation into the clinic, so to speak. So what led the charge next was hyperpolarized 13C MR spectroscopy. It still is a favorite technique for all the 13C MR spectroscopy for all purposes. And I'm not going to go into detail about DNP because I know everybody knows about DNP here. It's just basically you take a substrate of choice. In this case, it's usually pyruvate, and it's 13C labeled pyruvate, and you basically irradiate it with a microwave, a very strong microwave, as well as in presence of a free radical, basically polarizes your nucleus for a very small amount of time because the polarization relaxes pretty quickly. So you have only about a window of a few seconds before you can lose all that polarization. But that polarization increase gives you 10,000-fold increase in SNR. So you don't need too much substrate. You basically can acquire a very nice spectra, and you can also do 3D sort of image of your metabolites. So here's basically showing what hyperpolarized spectroscopy does. You have an initial dose of 13C pyruvate, and you're immediately starting to lose your polarization or 13C signal. And you see a turnover of lactate. It's forming, and then it's decaying also because of the 13C DNP being lost over time. But this amount of data is enough to fit your model to get kinetic rates of either the glycolysis or the neurotransmitter cycling, neurotransmitter energetics. So it's a very favorite technique to measure metabolism or metabolic imaging other than PET these days. And a very recent paper shows that basically. It shows that these are basically cancer patients who have been irradiated with radiation therapy, high-dose radiation therapy, and they're two groups, responders and non-responders. The only thing that's differentiating them is the higher lactate to total, carbon rate signal. So it means that higher glycolytic tumors have higher chances of survival to the radiation than the less glycolytic tumors. And we all know glycolysis is the primary, metabolic pathway that the tumors take, among many other things. But it's a very neat study showing that high glycolysis, tumors with high glycolysis have poor prognosis for radiation response, so to speak. All right, so moving on to another technique which was just developed, and these two papers basically arguably changed this MR spectroscopy field back in 2017 and 18, one coming from Minnesota, from Wei Chen's lab, and Yale, de Graaf's lab. So the idea is basically, instead of labeling glucose or your substrate with 13C, you have a deuterated compound, deuterated glucose. It's only labeled at 6 carbon because you lose all the protons in your glycolysis. So there's no point labeling it. Even if you do, you'll lose them. And then follow it up over time. So you basically, and acquire a DMR spectrum, which is much, much simpler. The deuterium has a very short T1 and T2. See, advantage is you can average very quickly within a few milliseconds, few hundred milliseconds, and the T2 is short. It goes against deuterium MR spectroscopy because your peaks are then very, very broad. So you don't have much spectral resolution. Nonetheless, you have no background other than the HDO, which is your baseline water in your tissue. And you inject the glucose, you see a 6,6 labeled glucose peak at 3.5, 3.4 ppm. And then later on, you start seeing the metabolites, GLX, which was, again, you can see from 13C spectroscopy as well as hyperpolarized 13C NMR. And you can basically, again, follow that curve, fit these data into the model, come up with your metabolic rates. So similarly, the other paper coming from Yale showed that you can now image, instead of doing an FID, you can image or 2D CSI, 3D CSI of your deuterium spectra. So here you can see in the healthy brain, so this mouse or rat has a tumor. And so you can see there's water in both the tumor voxels as well as healthy voxels, but you see a lot of glutamatergic metabolites versus lactate, which is a glycolytic product in the tumor. So it gives you basically a sort of an energy core of the tumor. And also you can implement it into the humans very easily because you don't need to inject, unlike 13C steady state. In the old days, when we used to do 13C spectroscopy, we can just drink this glucose and line the scanner for like 60 minutes or so. And we can come up with the turnover curves and nice metabolic maps of GLX, glucose, and so on. We can also, this is an example showing a patient with glioblastoma with high lactate in the tumor area. But see how the flare shows much higher hyperintense area compared to the lactic acid enriched area. That shows that this is basically probably the metabolic core of the tumor, which should be given more importance than probably the other tissues. So it's a very neat way of looking at the tumor physiology in 3D as well. So it gives you sort of a 3D map of your tumor and the brain as well, if you're interested in neurological applications and so on. And now more and more researchers are getting excited about this. Kevin Brindles publishes a lot of these studies in which he does in-cell NMR as well as a mouse as well as human, especially deuterium spectroscopy, showing the effects of therapies as well as glycolytic flux rates in these mice models. So this is an example of a flank tumor showing again the lactic acid turnover of the 6,6-2H2 glucose over time, and then you can measure the glycolytic flux rate from this metabolic model, and you can see post-treatment with the chemotherapy, just with chemotherapy, you can see that the glycolysis reduces. So you basically it's a sort of a biomarker of tumor response to therapy or not, and since it's really clinically easily translatable, it's given much more interest these days. Right, but you still require a specialized hardware to capture deuterium signal. So when we were reading those two papers back in 2018, 2019, we thought, why can we not do proton spectroscopy? And I'll talk to you about that in a minute. But just to get you up to speed about proton spectroscopy, it's basically, I know you've all heard about this, but it's basically the oldest MR approach other than MRI. I think they both evolved at the similar times, but it's a very simple approach. You have same NMR pulse sequence. You have a excitation pulse and then refocusing pulses with your slice selective gradients, and you get a nice spectrum from a brain region with all the metabolites. And since it's been evolving for like many years now, there are a lot of softwares that can actually process your data into individual components. You can basically see all the neurometabolites, both steady state as well as the ones which turnover change with therapy or whatnot, so to speak. So there's a recent article showing very nice study showing early on signs of decay in muscular MS. I'm sorry. And it comes apparently much earlier than the apparent anatomical damage to the brain. So there are many studies. So spectroscopy is catching on. People are also really interested in doing 2D MR spectroscopy, 3D MR spectroscopy with a very high resolution, with getting all the metabolite maps and so on. So we thought we should just do spectroscopy when we drink glucose, proton MR spectroscopy when we drink glucose, because what will happen, the glutamate which is getting labeled will not be apparent on proton MR spectrum because it's unlabeled now. So it will go down, and it did in the mice experiments that we did. And when we subtract the post-infusion spectra from the pre, we see an evolution of glutamate 4 signal only, and we compared that to the deuterium MR spectroscopy. Showed a nice correlation between deuterium MR spectroscopy, nice Altman agreement, and then the advantage we thought this offered over the DMRS is that we can, since we have much higher spectral resolution, we can actually look at different metabolites. So here's a curve for GLX, glutamate, glutamine, GABA, and then NAA, which is not getting labeled, so to speak. And then we also applied this technique into a tumor model. So what we have is a pre and post 6,6-H2-glucose spectrum from a tumor. And we can see that there's the 1.3 ppm peak, which is going down. It's a combination of lactate and lipid, but we all know that lipid turnover is much slower than the lactate turnover. So we can safely assume that this is coming from lactic acid. And so we can follow this up over time and generate a metabolic curve to again assess the glycolysis in this tumor. So the advantage of this technique, we think, is that it's easily implementable. You don't need a specialized hardware. Everybody has a proton MR spectroscopy, ways of doing proton MR spectroscopy, whether it's a head coil or any flex coil or whatnot. So it's easily implementable. But the problem with this technique is it's proton MR spectroscopy. And we all know proton MR spectroscopy is very sensitive to motion. So we cannot deal with a slightest of motion during the scans. Oh, by the way, there's another Nature Biomedical Engineering paper followed by our paper, which basically implemented this in humans on a 7 Tesla scanner, showing much higher resolution of the same glutamate 4 peak that we showed also, and a very good agreement with the 2H MR spectroscopy. And also they went ahead and they applied this on 3 Tesla scanner. So it's basically a clinical scanner where you can actually do QMRS on, and you can do a 3D sort of map following your GLX4 over time after drinking glucose. So at times you drink glucose and you lie in the scanner for 60 minutes, you have very nice, sort of decaying GLX4 maps that basically corresponds to your neurotransmitter cycling. So we thought that motion was a problem. We cannot do different spectroscopy always, because there'll be different space artifacts. So we thought we should basically train an LC model to basically incorporate the J coupling differences because of the labeling. So I'm assuming everybody knows what glutamate signal looks like. It has 3 different peaks at 2.1 and 2.4 and 3.7 ppm. This is coming from glutamate 4, which always gets labeled from the glucose that we are providing or drinking. So this is basically a glutamate spectrum from, when it's completely unlabeled, but then there could be a chance that glutamate will be also one of the protons will be labeled. So this is how the peak, the glutamate 4 peak would look like, and see how the coupling pattern between 4th and 3rd carbon is changing, and the 2nd carbon is unaffected because there's J coupling between 3rd and 4th carbon. And also when you have both the protons labeled, again, the J coupling changes the proton spectrum signal. So you can basically now, instead of subtracting the 2 spectra, because the patient will inevitably move during your scan, you can actually fit these spectra, avoid prior information to your processing tools, LC model, and you can actually come up with this nice decay curve of glutamate 4, glutamine 4, and other metabolites that you can think about getting labeled. And so you can basically draw a map showing the decay between gray and white matter, just as GLX4 after drinking glutamate, after drinking glucose over time. Okay, so this was basically my last slide, but this is a technique still in development. We see a lot of excitement about this approach, especially because it's easily implementable both in preclinical as well as clinical scanners, and it requires a lot of more research and effort into making it clinically available. But we're striving toward that, basically, if you have any suggestions or thoughts, please let me know either during or after the talk, this panel. But with this, I would like to thank you, and I would like to thank everybody who has contributed this, starting from Ravi Reddy's lab at UPenn, and Robin DeGraaf, and whoever I cannot think about. My lab at St. Jude, we are working towards this as well.
As always, give a word for our sponsors. First, MR Resources, they've been around for 39 years at this point, servicing the international NMR community and doing pretty much anything and everything that needs to be done with your NMR system. Magnet quench recovery, magnet and system moving and relocation, service contracts, reconditioned NMR systems, reconditioned magnets. And it's always very important for me to point out that they have absolutely a full supply of spare parts for everything that they sell. And they have engineering expertise that I dare say is second to none. Give some thought to doing business with MR Resources.
So for now, we'll continue with the second presentation. So I will introduce Dr. Isik. Dr. Isik serves as a faculty member at the Institute of Biomedical Engineering at Bogaziçi University. Her research focuses on developing new molecular MRI techniques to enhance the understanding of the biology underlying brain diseases. She has received funding from European Union, TÜBİTAK, and the Department of Defense for several research projects. And she's also an active member of ISMRM and ESMRB. So these are big international societies in MRI, so as similar to ENC, and she has served on those organizing committees. So in this presentation, Dr. Isik will discuss machine learning-based identification of genetic alterations in gliomas and meningiomas at the pre-surgery stage using multimodal MRI techniques. So with that, I'll hand it over to Dr. Isik.
Thank you, Dr. Sajja, for this nice introduction, and I would like to thank the organizers for this kind invitation. So today I'll be talking about our studies on machine learning-based detection of genetic mutations in brain tumors. And I would like to start with an introduction of my group, actually, because I will be showing you their work mostly. We are the Computational Imaging Laboratory, and we established our lab at the Institute of Biomedical Engineering at Bogaziçi University in 2016. Currently, we have 10 PhD and 4 master's students, and we have 8 females and 6 males, so we are a female-dominant group, maybe because I'm a female professor. This is our link for our GitHub page and our lab website if you would like to visit us. Our lab website is not up to date, really, but we are trying. I would like to start my talk with the MRI of brain tumors. And this is one quote that I really like. Irvin Hahn once said, "There is nothing that nuclear spins will not do for you as long as you treat them as human beings." And it is kind of true because we use the same MR machine and we could get information about the structure, also the vasculature and the metabolic information all together. And we don't even need to move the patient. It's very flexible and it's a great technology. When we look at our MR imaging categories, we have anatomical data for brain tumors, structural, vascular, and metabolic data. For the anatomical, we acquire usually pre- and post-contrast T1-weighted MR images. We have T2-weighted MRI and flare MRI. For the structural data, we get diffusion MRI. And for the vasculature, we have various techniques, DSC, DCE, arterial spin labeling, which doesn't need any contrast agent, and also susceptibility-weighted imaging. And for metabolic, we have the MR spectroscopy, which is, I think, dear to all our hearts. And there's also CEST and positron emission tomography. Let me start with anatomical MRI of gliomas. We could acquire, as I told you earlier, a T1-weighted MRI, pre- and post-contrast. I'm not sure if my cursor is visible. I don't have a nice cursor like Puneet, but I'm just going to use my, cursor. So we could get a contrast enhancing lesion after an injection of a gadolinium-based contrast agent. And this can leak into the blood-brain barrier disrupted areas, which are then going to appear bright. And this is usually indicative of a higher grade tumor. We could also get a T2-weighted MRI image, which has a hyperintensity in the areas of tumor, edema, and necrosis. So when we look at our conventional MR images, they have a poor definition of the tumor margins, and they are not quantitative. Plus, they can't provide any grade or tumor type information. Hence, we have our more advanced MR techniques, and perfusion MRI is one of them. It tracks the passage of a contrast agent through tissue in time, or we can use water as a tracer in the ASL MRI, and we can derive vasculature-related information. Let me show you an example patient. This is a T1-weighted image, and here's a malignant brain tumor. This is a DSC map of cerebral blood volume, and you see the high signal intensity at the same area where there is blood-brain barrier breakage. This is DCE, which gives us a K-trans map. Again, you see the hyperintensity. And this is arterial spin labeling map of cerebral blood flow. So they all show essentially an area where there is high vasculature. We could also use susceptibility-weighted MRI, which is a very powerful tool, and it provides high-resolution imaging of the vasculature. What we do in SWI is we get the phase information along with the magnitude image, and we multiply the phase map after some corrections, of course, because there's phase warping, with the magnitude image. And then we create the SWI image. We could also do minimum intensity projection of multiple slices, which shows a nicer view of the vasculature with a thicker slice. And in addition to the vasculature, our SWI images can highlight regions of hemorrhage, which appears dark in phase, and calcifications, which appears bright in phase. But I should note that this can be reversed based on the MR system that you use. Next is MR spectroscopic imaging of gliomas. We can obtain metabolic information from multiple voxels, in addition to the anatomical information that we get. Choline is a marker of cellularity or cell membrane breakdown. Creatine represents cellular energetics, and NAA is a neuronal marker. Choline and creatine are similarly leveled, and NAA is twice as high in normal tissue. In tumor, choline increases, NAA decreases. So this is a hallmark of brain tumors. And we could obtain actually quite a nice map of metabolic abnormality next to the normal voxels. I would like to note that if you were to look at the anatomical image behind this grid, it doesn't really show much, whereas spectroscopy highlights the tumor region really nicely. Next, I would like to mention about WHO CNS5. This is the new or latest, I should say, central nervous system brain tumor classification system, which was released in 2021, and it has advanced the role of molecular diagnostics. So the 5th edition of the World Health Organization classification of tumors of the central nervous system was released in 2021. And it builds on the updated 4th edition, plus C-Impact Now recommendations that happens to be in between the editions. And this WHO CNS5 has substantial changes that has advanced the role of molecular diagnostics in CNS tumor classification. What do I mean with that? Let's take a look at the table that they released. This is the gliomas, which is one type of the brain tumors, which is very common. And when we look at adult type diffuse glioma, currently there are 3 types. The first one is an astrocytoma IDH mutant. IDH is a molecular marker, isostrate dehydrogenase. And the mutation of this particular gene reflects a class. Next is oligodendroglioma IDH mutant and 1p19q co-deleted. And the last one is glioblastoma IDH wild type. So as you see, IDH is a major player now in the classification. And glioblastoma IDH wild type, which is the highest grade, now includes IDH wild type lower grades with features of glioblastoma. And what are those features? Again, those are mutations: TRP promoter mutation, EGFR amplification, and +7-10 copy number changes. So they are incorporating genetic information more and more in the classification of brain tumors. So IDH mutations actually are mostly seen in astrocytic and oligodendroglial tumors, and they are a marker of low-grade glioma biology. And I mark with green the survival curves of IDH mutant tumors. As you see in these two studies, actually IDH mutation results in a better survival and better treatment response than IDH wild-type tumors. When we look at the diffuse gliomas that do not carry this IDH mutation, they are usually glioblastoma, which is the highest grade. And now we have a glioblastoma IDH wild-type category only. Additionally, if we don't have IDH mutation, but have a TERT mutation, those patients are called TERT only, and as marked with this red arrow, they have the worst overall survival. I'm mentioning these because these are actually the mutations that we used in our studies. There have been some key findings in terms of IDH mutation markers, and IDH mutant gliomas have higher 2-hydroxyglutarate, which is an oncometabolite. They have lower glutamate and lower glutathione. We could also create 2-hydroxyglutarate maps for the whole tumor region. Choi et al. Released this study actually in 2012, and they actually developed this optimized press sequence with a TE of 97 milliseconds. Within the tumor region, you can see 2-hydroxyglutarate. When you go to the normal part of the brain, there is no 2-hydroxyglutarate. So you can create actually 2-hydroxyglutarate maps, just like your choline maps and NAA maps to indicate the tumor region. Next, I would like to briefly mention our work in radiogenomics and machine learning at our lab. Molecular alteration information that I have mentioned so far could only be obtained through invasive biopsy or surgery, and there's a need for non-invasive preoperative molecular profiling. And that has actually caused two of our grants. The first one was, these are all national grants, by the way. TÜBİTAK is our NIH in Turkey. The first one was aiming to create machine learning methods to preoperatively predict IDH and TERT mutations in gliomas based on multimodal MRI. And our second grant actually focused on deep learning to preoperatively predict NF2 mutation and S100 immunopositivity in meningiomas based on multimodal MRI. And I will briefly mention some of our results based on those two grants. And our collaborators were from Acıbadem University. In our first publication, we showed that we could identify IDH mutation and we could differentiate IDH mutants from IDH wild-type tumors by using machine learning with 88% accuracy. And this was an MR spectroscopy work. And in the primary gliomas, again, our accuracy was similar. And when we looked at TERT mutant tumors within IDH wild-type group, which is actually your TERT only group, which is the highest, risk group, we identified them with 92% accuracy. And these are the selected features we used in our studies. And here's an example IDH mutant grade 3 astrocytoma patient, that's TERT wild-type. And here's a glioblastoma, higher grade patient, that's an IDH wild-type patient. This blue line is 2-hydroxyglutarate. It's not very visible. These are LC model outputs, but you don't see it in the IDH wild type versus there is the 2-hydroxyglutarate peaks in the IDH mutant patients. When you look at inositol, it is higher in the lower grade. Again, choline creatine levels are similar. The choline is higher in tumor, NAA is lower, but in the higher grade, we also see high lactate. We created this open source glioma genetic diagnosis tool, which could actually predict IDH mutation based on the levels of the metabolites. Of course, this is trained on our data only. It is not really generalizable possibly. We have to work on it. But, based on the information that the model has so far, it predicts if the patient has an IDH mutation or not. It can also allow you to depict, the data. Plus, we have some visualization tools and an advanced user module that you could create your machine learning models and also use feature selection and data augmentation. Abdullah did this study, and this is our GitHub link if you would like to check it out and provide some feedback, which will be awesome. We applied a one-dimensional convolutional neural network, which is a deep learning method to detect IDH and TERT mutations in gliomas using proton MRS. And based on our results, actually, we included 237 patients, which was a lot of actually patients for this particular study. And we identified IDH mutation with 94% accuracy. When it comes to TERT mutation, it is not as clearly defined in metabolism. And we only achieved a 77% accuracy, and we identified TERT-only patients with 82% accuracy. We also used susceptibility-weighted MRI that I showed you earlier. We combined FLAIR, SWI, and contrast-enhanced T1-weighted MRI from the same slice. We cropped it to a tumor region, and we created an RGB image, as we call it. And we fed this into a deep learning algorithm and a machine learning algorithm. Their combination using a deep learning algorithm resulted in 92% accuracy, actually. We also used perfusion-weighted MRI. We took the whole tumor from the T2-weighted image and necrotic core from the T1-weighted image. And we registered these maps onto the RCBV image. And we selected the tumor region. We either used these three, different techniques, T1, T2, and RCBV, and created an RGB image out of it, or we took the biggest tumor slice and the next and the previous slices and created an RGB image. And we fed it into a pre-trained network using transfer learning. As a result of our training, we were able to identify IDH mutation with 88% accuracy. 70% accuracy was achieved for third mutation, and 84% accuracy was achieved for third only detection. Next, I would like to briefly mention meningiomas and our second work. Meningiomas actually comprise 36% of all primary brain tumors, and there's a loss of neurofibromatosis 2 function that has been observed in 2/3 of the meningiomas. So these NF2 alterations have been associated with a more aggressive tumor biology, and these patients tend to have a shorter time to recurrence and shorter overall survival. There is also S100 protein expression, and on the contrary, this is a good indicator, and they tend to have lower WHO grade and better prognosis. So our aim was to preoperatively determine NF2 loss and S100 protein expression in meningiomas using deep learning. We applied 1D CNN for grading of meningiomas using proton MRS. We used 57 meningioma patients, and we developed a one-dimensional CNN network. And our accuracy was around 81% for finding the higher grade meningiomas. We also identified NF2 loss in meningiomas using proton MRS at 3 Tesla, and our machine learning, classical machine learning achieved 77% accuracy, whereas our 1D CNN model achieved 89% accuracy. We also used susceptibility-weighted MRI for S100 protein expression prediction. I don't even need to talk. Take a look at these two images. This is an S100 negative, and this is the S100 positive data. In the S100 positive, there is much less vasculature. And as I told you earlier, this is a lower grade marker. As a result, our median values for the S100 positive group was higher because they had less dark spots. And we were able to predict S100 protein expression with 85% accuracy out of susceptibility-weighted MRI. So, where are we in the clinical translation of machine learning? We have been developing several techniques, and we are not, of course, the only one. There are a lot of groups out there, but there are a lot of challenges in translating machine learning into the clinics. First, data quality is not great. Most of the time, it's incomplete and noisy. There is lack of big data. People don't want to share. There's imbalanced data, so, it's difficult to detect rare cases. We have generalization issues. I can create a model, it might not work in your hospital. Explainability is a problem because usually these deep learning models are black box. There's also regulations. In Europe, we are bound with the MDR regulations, so we have to be careful about, releasing anything out into the clinic. There's data access and ethical issues. And there's acceptance and training because, medical professionals might not be willing to accept your solution and use it in their clinics. So there are a lot of issues, actually. Let me tell you what we have done for explainability, which is only a few slides anyways. We applied GradCam. This is a technique, gradient-weighted class activation mapping, that shows you where a model focuses when it makes a decision. So here where there is, low signal in SWI, actually, your model focuses on that particular region to predict S100. And in this particular case, the area under the curve was 84%, and GradCam really nicely highlighted low signal intensity areas. We also used it for RCBV maps. And in the third only group, as you see, our heat map is focusing on the whole tumor region, especially the edema. And in the IDH wild type, on the contrary, it focuses on the necrotic core. For MRS, actually, it nicely picked the important peaks. As an example, in the IDH mutation, it picked the 2-hydroxyglutarate. Data sharing is another problem, as I told you. We need some solution, and federated learning has been one of the solutions. I'm about to finish, by the way. Here's an example of federated learning diagram. You can train 2 models in 2 separate hospitals, and then you can aggregate model parameters using another server that's central. We applied federated learning on UCSF PDGM dataset. This is an open dataset with 501 glioma patients, and we achieved 93% accuracy in centralized learning and up to 89% accuracy in federated learning. So the use of machine learning in clinical applications actually requires a multidisciplinary approach. We need professionals from different fields. Engineers are not enough. We need doctors, ethics experts, law experts, and we need to collaborate. And we need a rigorous testing and evaluation process for the development and validation of machine learning models. And proper calibration of this technology is necessary. We need to consider data privacy and ethical issues. Federated learning is a solution for data sharing, and it is important that medical experts verify and interpret machine learning solutions. In conclusion, actually, molecular markers play a significant role in understanding glioma biology and meningioma biology, and they have been more increasingly used in WHO brain tumor classification. These are not random molecular alterations that occur by chance. They actually indicate a specific tumor biology, and machine learning has gained a popular role in preoperative classification of brain tumors. And there's still a lot of room for improved deep learning approaches, improved studies, generalization, robustness, explainability, and reliability are all issues. I'd like to acknowledge my students who have contributed slides to this presentation and our collaborators at Acıbadem University and Siemens Healthcare and these 2 grant supports. And I also would like to thank you for your patience and interest. Thanks.
We invite you to get to know Q1. Q1 is dedicated to developing NMR systems from 400 to 600, as well as full system upgrades from 300 to 600 as well. You can get a lot of features in a 2-channel NMR spectrometer at a great price. So we invite you to get to know more. Contact us at q1americas.com or info@q1americas.com by email. Thank you.
So I will let Esin, so I'll be the last speaker. I'll be talking about liver MRI, and I'll let Esin introduce me.
Thank you, Aaryani. So Dr. Aaryani Tipirneni-Sajja will be our next speaker, and she's an assistant professor in the Department of Biomedical Engineering at the University of Memphis. Dr. Sajja also holds a secondary appointment in the Department of Diagnostic Imaging at St. Jude's Children's Research Hospital. Her research focuses on developing quantitative and automated MRI and NMR spectroscopy methods that will advance the patient care. And she has secured research funding from NIH as a Trailblazer Award and received NSF Career Awards and several university grants. And she will discuss MRI techniques for assessment of iron overload and fatty liver disease. So, Dr. Sajja.
Yeah, thank you, Esin. And I would also like to thank Ivan NMR organizers for inviting me, especially Dr. Krishnamurthy for inviting me to lead today's workshop. And yeah, today I'll be covering about quantitative liver MRI. And the last thing I would like to add is in 3 days, I'll be starting as associate professor at University of Houston. So yeah, I got my tenure, so which is good. All right, so I'll just show, this is our lab. So what we do is we do both MRI and NMR metabolomics research. So if you think of NMR, I mean, I know you're from the NMR field. So me, my postdoc, my PhD and postdoc was all in MRI. And I also started doing NMR metabolomics work after I started as a faculty. So I'm coming from the MRI world. I mean, I went to the NMR, and we can do vice versa. From NMR, you can also go to MRI because it's basically the same principle of magnetic resonance. In NMR, you're acquiring a 1D spectra. In MRI, you're acquiring a 2D image. And you're doing like spatial localization to acquire your image. And if you are interested in knowing more about our lab research, you can go to this website. All right, today, the outline of my today's talk would, I'll briefly cover over the liver pathologies, so iron overload and steatosis, and how MRI can be used to quantify iron and fat. And lastly, I'll touch upon my lab research as a big picture. So liver iron overload, it's a problem in patients with hereditary hemochromatosis where they absorb too much dietary iron and end up with iron overload. And liver is the primary organ for storing excess iron. And it also plays a main role in iron homeostasis. So, and iron overload can also be acquired in patients with blood disorders and cancer who receive chronic blood transfusion. So if you're injecting blood in these patients with hematological diseases, so each unit of blood is about 250 milligrams of exogenous iron. So these patients receive chronic transfusions. So if a sickle cell patient is on 6 months on transfusions and receiving 2 transfusions per month, ends up doubling his normal iron content in about 6 months. And all this extra iron is stored primarily in liver, and also it can store in heart. And the body doesn't have a natural mechanism to remove this extra iron. So biopsy is a gold standard to know how much amount of iron is there, and then prescribe chelation dose to the patient to remove the extra iron. So we need to have a tool to monitor how much amount of iron is there so that the hematologist can prescribe the chelation dose to remove the extra iron, because extra iron is toxic. But biopsy, as you know, it's invasive, it's risky, and the patients can have risks such as bleeding and infection and other complications. And over the last 2 decades, MRI has emerged as a non-invasive measure to quantify how much amount of iron is there. So if you see this, these are the biopsy images. So these are the histology images, and the blue stain here is showing the iron. And this patient has like a mild iron, and this patient, you see a lot of blue here. So these are all hemosiderin deposits. And it's saying that the patient has too much iron and needs to be treated. And if you see, these are the corresponding MRI images. And what you notice here, so you can obviously see the liver is bright on this image, whereas the liver is dark on this image. It's because the iron would distort the MRI signal and making the liver look darker. So how can we use MRI to actually quantify iron? So this is a patient with normal HEC, so which is hepatic iron content. So I'll use these abbreviations. So if you see the signal decay in a patient with normal HEC, so the signal decay looks like this. Again, this, this is a FID signal, which you acquire in NMR. But now if I take cases with more iron with moderate and severe, you see the signal is decaying rapidly. It's because the presence of iron creates local magnetic inhomogeneities and causes the protons to dephase faster. So because it increases the spin-spin relaxation rate. So, and you can see the signal decay is much steeper. So you can actually quantify this the rate at which it is decaying and use that to estimate how much amount of iron is there. So the higher the iron content, the signal decays faster. So you should be knowing about T2 star. So T2 star is the transverse relaxation time. And in MRI, we use R2 star, which is just the reciprocal of T2 star. So R2 star stands for transverse relaxation rate. And the reason I'm using R2 star is the more the iron, the higher is the R2 star. So it's a very linear relationship. So just to give a basic, I don't know how far you know about MRI sequences. So we use a GRE sequence. I think GRE should be familiar with you, but the only difference for creating an image is you have this gradient localization. So you use a slice select gradient, a phase encoding, and a frequency encoding gradient. So, and you fill each line in K-space. And in MRI, you use echoes rather than FIDs. So what the gradient recall echo is doing here is this is your FID. We apply a dephasing and rephasing gradient to form an echo. And this echo is used to fill each line in K-space. So all the raw data is stored in the K-space. So each line, so you use a different phase encoding step each time and fill in the K-space. And after filling in the K-space, if you do a 2D Fourier transform, you get your image space. So it's basically you're going from your FID to spectra. So here we are going from K-space to the spatial domain, which is the image. So the sequence we use for estimating the R2 star is a multi-echo gradient echo sequence. So these echoes, we are having multiple echoes. So we are using a dephasing and rephasing gradient to produce these echoes, and we are capturing these signal time points. So this is an example showing, so that these are the images, these are the 20 images obtained. And what we are doing is we are capturing the signal. And if you see here, this is the signal over time. And the rate, and this is, as you see, is a mono exponential decay. And you can quantify the decay rate using the decay constant T2 star. And again, as I said, the reciprocal of T2 star is R2 star, which is the rate. So you can actually quantify at which rate the signal is decaying. And this is the signal curve for a patient with normal HIC. And this is for the moderate HIC, and this is for the severe HIC. And as you see here, the more the iron, the steeper is the decay. So the shorter would be your T2 star, or the higher would be your R2 star. Okay, now we got R2 star. How can we translate this to hematologists or the clinicians to tell? Because R2 star is an MRI measure. It's not the iron content directly. Early studies in 2005 and 2009, what they did is they calibrated the R2 star values with the biopsy HIC values. So they actually measured the iron content from biopsy using mass absorption spectrometry. So that's a gold standard. So they measured the iron content and they correlated with the R2 star measurement. And if you see here, it's showing a very excellent correlation. So they developed the calibration equations where from the MRI, you can quantify the R2 star, plug into this equation, and then tell how much amount of iron is there so that the hematologist can use this iron content to prescribe the chelation dose. So typically the normal iron content, it is less than 1.2. It's normal. If it is between 3 to 7, you classify the patient as mild iron overload, 7 to 15 as moderate, and more than 15 as severe iron overload. And the chelation dose is different for each of these categories. So it's very important, it's important to know how much amount of iron is there to prescribe the accurate chelation dose. But the limitations of MRI are in case of high iron overload, as you see here. So this is a Bland-Hartmann plot showing the mean hepatic iron content and the hepatic iron content that is estimated using R2 star with reference to biopsy. So as you notice here, for severe iron cases above 15, you see there's a large deviation. So you see there's loss of precision in the measurements. There's not good agreement between R2 star based iron measurement and biopsy measurement. And the reason for this is, as I said, the signal decays very fast. So this is again the GRE sequence, what I told. So the earliest echo time we can achieve with the GRE is 1 millisecond. So in cases with patients with severe iron overload, the signal is already decayed. So the first time point you see here, so you only get the first time point, but the rest of the signal points are almost in the noise floor. So because of this ambiguity, so the precision is lost. And an alternative technique for this is ultra-short echo time imaging. So where you can go to as low as 0.1 milliseconds. So again, UT is a version of GRE, but it uses radial sampling, which I'll show in the next slide. But what I want to highlight is with the UT imaging, you can go to much shorter echo times and acquire more signal points so that now you can fit the curve and you can measure the R2 star accurately. So again, if you compare, this is the GRE pulse sequence and the UT. So the GRE, it uses a traditional Cartesian-based sampling. So where you fill K-space line by line, whereas in UT, you go with radial. And here with UT, you're doing center-out radial sampling. So you use half RF excitation with alternating slice selection gradients, and you fill your K-space. So instead of filling them like this, you're filling them as radial spokes. So you fill all of these, and again, this is your K-space, and if you do the 2D Fourier transform, you will get your image. And as you see here at the center, the another advantage with UT is you're oversampling the center of the K-space. But whereas at the periphery, you're undersampling. So what does it mean? Again, You're coming from NMR, I'm not sure how far you know. So a quick thing about MR image formation. So as I said, K-space, we save all our raw data in K-space. And in K-space, the center has the low spatial frequencies, whereas the periphery has the high spatial frequency. So this is your K-space. And once you apply the inverse Fourier transform, you get your image. So all the data we capture is in K-space, and then we have to apply the Fourier transform to see the actual image. So what that means is the center has the low spatial frequencies and it gives the basic image contrast, whereas the periphery would add more resolution to the image. So why I'm saying all this, in UT, we are acquiring more at the center and less at the periphery. But what it is with this, the advantage is it will help to be motion insensitive. Because of the oversampling, you're averaging the motion. So again, in liver world, in abdomen, so the patient has to hold the breath to get the scan, because if you're breathing, the breathing motion would cause motion artifacts. So this is an actual example explaining that. So for abdomen MRI imaging, the patient has to hold the breath. So this is a standard GRE, multi-echo GRE sequence. The quality is very good, and this is the R2 star map. And now if for patients who are unable to breath hold, or in sedated patients who are sedated and cannot do the breath holds, so you see all these motion artifacts. So these are all respiratory artifacts, and these would affect your R2 star quantification. So if you see here, so all these are causing underestimation of R2 star. And this is the advantage of UT now. So apart from enabling us to quantify high iron cases, so it's also helping so you can also implement the sequence in free breathing cases. So this is a case again where it's a free breathing UT, and you see it's not, the motion artifacts are not that apparent. The resolution is poorer because you're not collecting as much in the periphery. But if you see the R2 star values, they're pretty close to the standard breath hold GRE. And you see, this is one more example in the sedated patient. This is the same thing. So this is a free breathing GRE, and you see all these artifacts causing R2 star underestimation, and it would affect the iron quantification. Whereas UT, you see only minimal streaking artifacts because of radial sampling, but the R2 star values are still reliable, and you can see the vasculature very clear. All right, so let's switch gears. So that's the iron part, and I will show at the end what we are doing in iron and fat research. But the other important function of the liver is fat metabolism. So as you know, the liver, it oxidizes triglycerides, and it converts all the excess carbohydrates and proteins into fatty acids and stores it here. And fat accumulation, which is called steatosis, is a huge problem. It's the hallmark of non-alcoholic fatty liver disease. So if you see this is the normal liver. And because of the sedentary lifestyle, the high-fat Western diet, the insulin-resistant obesity, all these factors would lead to non-alcoholic fatty liver disease. And in recent months, it was renamed as metabolic associated fatty liver disease. So the problem with this is asymptomatic till here. So once you start fibrosis, it leads to cirrhosis, and then it will lead to liver failure. So you'll have to monitor it early to make it reversible. And this is showing the prevalence of non-alcoholic fatty liver disease. And you see, this is a few years back. I assume the numbers will be higher now. But if you see 1/4 of the North America, and also Middle East, Europe, everyone has more prevalence of non-alcoholic fatty liver disease. So this is really a big problem. And again, biopsy is a gold standard. You see here, these are the histology images, and here these white circles here are showing the fat deposits. And again, we don't want to do biopsy. So MRI has emerged over the last 2 decades to measure fat, so which I'll explain in a second. So again, the standard is MR spectroscopy. It's the oldest technique. So if you, again, this is very similar to NMR, as you know. So where we place a voxel, and then we measure the signal, we acquire the signal for so long time, and we apply Fourier transform to get the spectra. So if you see, I mean, the spectra is not as good as your spectra, right? So this is because we are capturing at 3 Tesla and your guys are capturing at 9 Tesla or 11 Tesla. Anyway, so this is the water signal. This is the in vivo MR spectrum of a fatty liver disease. And all these 1 to 6 are the fat peaks. So this 5 is the methylene peak, which you might be knowing. The 6 is the methyl peak. And the 1 is the olefinic peak and so on. So this is the triglyceride spectra. So what we do in MR spectroscopy is we measure the fat area under these 5 or under the 6 peaks and divide it with the water area plus fat area to estimate the fat fraction. So again, this is in vivo you're doing. But the problem with MR spectroscopy is you're losing all the spatial information. You're only restricted to this one voxel. And the other problem is iron. So in situations where the liver has both iron and fat, the iron would cause these peaks to broaden, which I'll show in the later slide, and you cannot quantify the fat fraction. So in imaging, we also have this in-phase and out-phase imaging where you acquire images. So if you come to the MR world, so when you acquire the multi-echo images, because the fat and water resonate at different frequencies, they come in phase and out phase. So for example, this is at 1.5 Tesla. So they're out of phase at 2.2 milliseconds and they come in phase at 4.4 milliseconds. So what this is doing is because of this, it creates oscillations in your signal decay. So if you acquire a GRE signal, you see these oscillations. So the high show is when the fat and water come in phase, and the low is when the fat and water become out of phase like this. So we can use this to quantify the fat fraction. So you subtract the in phase from out of phase and divide, and you can actually get the fat fraction. But then this is called the 2-point Dixon method. But then the problem with this one, it doesn't account for the signal decay that is happening. So I showed you, right? Even for the normal liver, the signal decays over time. So the in phase, out of phase wouldn't account for the signal decay. And also in cases of coexisting iron, there would be a large bias in the fat fraction. And also this in phase and out of phase, what you're seeing is mainly from this methylene peak, but the fat has the other peaks too. So yeah, it's not accounting for the multispectral fat complexity. So this is a case that has both iron and fat. And if you see, so these are the oscillations, but because of iron, the signal is steeper. But now if you fit like a mono exponential signal decay, then you might not get an accurate R2 star value because of these oscillations. So for this, so over the, I think, last few years, the fat water R2 star modeling has come up, which uses nonlinear least square fitting. So this handles both iron and fat. So you can use the signal model to estimate both iron and fat, because iron confounds fat measurements and fat confounds iron measurement in MRI. So you need to take both into account to quantify both accurately. So again, this is the FF is the fat fraction, and R2 star is the one that would determine your iron content. So as I said, there are challenges in the concurrent presence of iron overload and steatosis. Iron would confound fat fraction measurement, and fat would confound R2 star. And also there are cases, diseases where both can coexist. And if you see here, the problem you can see, these are the signal decays, and you see the higher the R2 star, you don't anymore see the oscillations. This is a fat fraction with 20%, and you don't see the oscillations when there is iron here. And this is in time domain. But if you see in frequency domain, as you know, so iron would broaden the peaks. So this is the high severe iron case of R2 star 1000, and you see this is your water peak and this is your fat peak, and you see one as this big lump. And it would not allow, so the spectroscopy also would fail in both iron and fat situations. So I'll just touch upon my lab research. So we are kind of trying to answer these. We are trying to develop a model that can quantify both iron and fat accurately. So we are developing MRI techniques. We are using simulations, phantoms, and finally validating in patient data. And again, this is funded by the NIH. So what do I mean by simulations is we are creating our own virtual models because it's hard to get biopsy samples. So we are creating these iron overload models, and these are the synthesized MRI signals. And this is the fat model, and we are synthesizing the MRI signal. So what we did, again, I'll briefly run over it. So we took the histology. I mean, this is the work that was done by Gugre et al., which was very helpful. So he actually quantified the morphology of the iron deposition, so the size and distribution of iron deposition. So he did this on histology images, on microscopy, and then he estimated the size of these deposits and the distribution. So we used all these parameters or morphological characteristics to develop our own iron overload model. And if you want to know more about this, you can go to the publication, but this is the model. And then we created the signal, and we also verified the relationship between R2 star and HIC if it is similar to the in vivo behavior. So I showed you the calibrations in vivo. So if you see here, the blue dots here are from our model, and they pretty much agree with the calibrations, with the Wood and the Hankins calibration. That means showing that our model is realistic to in vivo deposition. So similarly, we built our steatosis model, but for this, we didn't have previous studies that actually characterize the morphology of steatosis. So with this, we had to do by ourselves. So we actually collected all the histology images, quantified the fat droplets, the size and the distribution. And then, so again, these are the different parameters we quantified. And then we created our own steatosis models from the histology. And these are the synthesized MRI signal, and we investigated our model behavior using these simulated data. And lastly, I mean, we also are interested in investigating the low field, because as the field strength increases, the signal decay would be more rapid with the presence of iron. So we were testing if the low field would slow down the signal decay if we could accurately quantify both iron and fat at low field. And our preliminary results show that at 0.55 Tesla showed improved fat fraction estimation bias compared to the 1.5 Tesla here. So again, we are also investigating if low field really helps here. And we are similarly building our own phantoms. Again, these are all simulations, right? How real they are. So we are also building our phantoms with fat and iron. So we are using peanut oil for mimicking steatosis, and we are using iron particles of different sizes. And we are also doing validation to make sure the phantom micrographs are similar to the biopsy sizes. So the fat droplets are similar in size as the biopsy fat droplets, which are very similar. And we also saw that the iron particle size affected R2 star and fat fraction. So I'll briefly go over it. But what we observed is the smallest iron particle had high R2 star, even for the same iron content. So which we thought was interesting. This is still work in progress, but it shows that the iron particle size would also affect R2 star and fat fraction quantification. And this is also showing that the smaller iron particles had more fat fraction bias compared to the larger iron particle size. And lastly, I mean, we're also developing some deep learning techniques to automatically extract the liver, because clinically, the radiologists have to draw these ROIs and then quantify. But we are also developing a deep learning technique to automatically extract the liver, filter off the blood vessels, and quantify the mean R2 star. And this is a work in progress. Our final aim is to fully automate the parenchyma extraction and also the reporting of iron content and fat on the MRI console itself. And with that, the last slide, the last two slides, I also want to say, so we do NMR metabolomics research, so where we are interested in developing quantitative and automated NMR techniques. So this is a recent paper, so we're interested in developing deep learning techniques to automate the quantification. It's similar to Esin, what she's doing, but in NMR. So where we are inputting the NMR spectra, and then it spits out the concentrations of the metabolites. So again, this is the proof of principle paper. So if you're interested in more, we validated in complex mixture experimental spectra and also tissue samples. And we got very good, interesting results. And a plugin, so we have this huge project, very ambitious, developing a metabolomic software for high field and benchtop NMR. So as you're all NMR audience, so if you're interested, please feel free to reach out to me for any opportunities. And with that, I would like to thank my collaborators at the University of Memphis and my outside collaborators at St. Jude and Northwell Health. And this is my wonderful lab. And without this, all this work couldn't have been here. And I would like to also acknowledge my funding resources. And with that, I'll stop here and take any questions. And now the panel is open for discussion.
If there are questions, you can unmute yourself and ask the panel directly.
Yeah, please feel free to ask from any of the presentations because we wanted to finish the presentations and leave more time for questions.
Puneet, this is Krish. I was sort of one of the. I'm a very novice to imaging, obviously, but you mentioned something about the increase in the as the profile or the intensity increases, and the inflammation is a lot more in the initial part of a particular signal in time. We see that a lot in liquids, particularly in reaction monitoring. In the kinetics, it's the transition time that has some best information. And one of the tools that we built, again, it's a high resolution liquids, is what we call a Mystique model in situ spiking. So essentially, you can take, because it comes from the same reaction, you can take a signal from another time point, model it, and use that model to digitally spike the entire set, and essentially increase your detectability limit or amplitude. You can call it amplitude decrease amplitude uncertainty at the very low levels. And that seems to fit significantly better by simply taking post-acquisition, obviously, taking the model from more intense time point and spiking that model mathematically to the entire set, and then fitting that set, assuming that you just increase your intercept of any fit that you try to do. That has significantly improved the way we would characterize the kinetics part of the reaction. So I just wanted to post that and then see if there are things that you have tried. In the imaging world, there is a lot of modeling tools. Can you mathematically improve your quantification at the very early part of your C13? I think I saw that in your C13 glucose study that you showed, that at the early point that it's noisy. So that's just wanted to point out that perhaps there is a synergy between the problems we face in the liquids area and the problems you are seeing in the imaging area as well. It's just a time versus amplitude. Yeah.
Yes, Krish, that's a great suggestion, and I think people, I completely agree with you. First of all, and I think people have already tried it in 13C field. I remember reading a paper from Murli Sharkuri's group. Jeffrey Brender, I believe, was the first author, in which they tried denoising the 13C spectrum and coming up with using AI machine learning tools. But yes, it's basically exactly what you're talking about. So yes. I mean, also the QMRS or Quelt MRS that I presented can also be helped a lot with AI machine learning.
Our modeling approach is primarily based upon Bayesian fittings of the time domain data. So.
Right.
I have a general question. if I could ask it. I know very little about AI, but what kind of software do you, in general, take up when you start making these deep learning models?
Maybe I can answer. There are available tools, lots of Python libraries. MATLAB provides some AI tools as well, but I think Python has taken it over. And there are lots of actually AI libraries that one can start off with. Usually it's a matter of optimizing it, fine-tuning it, and then there are also available models that one can, adapt to the particular problem that we are solving. As an example, there are lots of models trained on ImageNet. It's a big data set full of images, but regular life images like butterflies, trees, cats, dogs. But the model learns a general representation of the image. And then you can take it and then you can apply a model that was trained on ImageNet for a particular radiological problem. There is also this new data set for radiology images, which was published recently. So what I mean is, Python and MATLAB, those are available. There are also tools that will use these that are already developed. Lots of data out there that can be, taken and then applied a model on. But overall, we still lack lots of things like, we don't have big data sets. That's one thing. We don't have very big data sets for our particular problems.
So these are open source information, and Python is an open source, is a freely available program.
Freely available.
So really anyone could get started without too much investment.
And there are a lot of good courses on Udemy, Coursera. We are all following it. So lots of resources, definitely.
Okay. Thank you.
Thanks.
I guess if I can jump in, I had a general question for Aaryani. It's kind of very general, because again, I'm not in imaging either, but I'm very interested in the liver. And so in terms of the fat content and the R2 measurements, maybe you address this. How much are these affected by the proportion of the macro versus the micro vesicular droplets? And how do the relative proportions factor into, for example, R2?
So I think, so MRI, we are only speaking about macrosteatosis. I don't think it's that sensitive. So it's primarily macrosteatosis more than at least 5 micrometer. Yeah, that's what we are looking at. Okay.
Mm-hmm. Thank you.
Sure. Thanks for the question.
May I ask a question to Aaryani?
Mm-hmm.
Very cool work. Amazing, really. And I was amazed with the UT and how insensitive it is to motion. I was wondering about if you have tried any particular, fast imaging techniques, because I know that radials are quite insensitive to parallel imaging or, fast MRI applications. Have you ever tried that?
No, we haven't. But so even with this, the UT I showed, I think it's a 1 minute scan. So even we got good results with just one average, whereas with GRE, if you're doing the similar averaging, it's comparable. So you still have to do like a 1 minute imaging to get at least 3 or 4 averages. So yeah, we haven't explored that yet. But there is also from vendor side, a lot of radial GRE coming out, which is breath hold. So those are Siemens has work in progress packages for radial GRE.
I see. Another question that I had was, have you tried multi-compartmental R2 star feeding? Like, I see that there is vasculature and there will be some blood contribution in some of those voxels. So have you tried by exponential fitting?
No, because I mean, we try to exclude the vasculature. So once we either do the whole liver ROI or the small ROI, we try to be away from the vasculature, but that would be an additional fitting variable, and you might not get, it might, too many fitting variables might again reduce the stability of your model. So, but that's a good question. I know R2, like FerriScan, they do bi-exponential fitting. So for slow and fast decaying R2.
I see. Thanks, Aaryani.
So Esin, I have a question for you. So I was wondering, what were your inputs for the machine learning thing? So was it the entire spectra you're giving, or is it only the concentrations of those metabolites?
The whole spectrum, Aaryani. And
Okay.
in some deep learning models, we were providing the fitted model, the baseline, the whole spectrum, all those. And then we were feeding, into that data. We weren't using, for the classical machine learning, we first quantified our spectrum using LC model and then used those for classical machine learning. But for deep learning, we provided the whole spectrum.
Okay. But you're not quantifying the metabolites with deep learning, right? It was only for classification of the...
Exactly. We weren't quantifying the metabolites. That's another actually area of research. And I think you have done a similar something that looks very cool.
Yeah.
I will read the papers.
And one last thing, so as a follow-up question, but so the ground truth you're having is the biopsy classification?
Yes.
Okay.
Ground truth comes from biopsy, and a histopathologist evaluates the IDH mutations, TERT mutations, or the other mutations in the patient data. And we look at the data retrospectively. These are the preoperative data that was already collected after the surgery. They do the biopsy and then, or sampling, and then they look at the mutations. And we kind of, correlate pre-surgical data with the post-surgical mutational information.
Got it. Okay.
Yeah.
Yeah, that's cool. And I also like the SWI thing. So those are in humans, or is it in... oh, wow. So what is the resolution?
It's higher resolution, but I don't remember the matrix size. It's better than C or DC. I could check and let you know.
And what field strength?
Those are all 3 Tesla.
Oh, 3 Teslas too. Yeah, they look very good.
The example that I showed in the first few slides was from 7 Tesla.
Okay.
But the ones that we used were 3 Tesla.
Okay.
Can I ask a question to Puneet also?
Yes, please. Esin.
Very cool work, Puneet. I had no idea, this could be possible, actually. There was only one thing that I was kind of surprised at. Your scan time. It goes up to like an hour, right? So how do you keep the patients? Do you scan them, take them out, and then put them back in? Or how does it work? I was wondering.
Yes. For the mice, it's easy. They're always anesthetized.
Yeah.
For the humans, we tried a different approach than the Vienna group, which I like a lot. So what we did is we did a baseline either a day before, or before doing the glucose uptake, and after the glucose we had 60 minutes. Whereas in the Vienna group, they started their baseline just after drinking the glucose, and they followed it up for like 60-70 minutes. Yeah, invariably you have to do a long-term scan to be able to do the dynamics, to measure the dynamics of the turnover. But I guess we can space it out. We can, if we like you were saying, with more deep learning AI models, we can actually do with lesser amount of spectroscopy, maybe some just initial, as Chris also suggested, some initial spec data that'll put us on the path of like initial turnover rates. Whereas, steady states data will also be valuable, but not as much as initial turnover rates. So, right now, as it stands, we need whole turnover data, but over the time, this problem can be solved with AI machine learning problems. And also, I would like to point out that, like you said, there's also going to be emotion because it's a long scan. So that's why we tried to avoid the difference taking the difference between the baseline and the post, glucose uptake scan, intake scan, and we are training LC model to, or basically we're training basis sets in the LC model to be able to, pry information about the J-coupling differences based on the labeling patterns that we would observe.
Thanks.
Thank you. Thank you for the question.
So Puneet, so you talked about deuterium. So taking deuterium is safe?
Yes, actually, people take a lot more deuterium than we did in this because there are liver studies which you'd be interested in, which measure lipogenesis in the liver using deuterated water, and that's a lot of heavy water. And they have to drink for like the whole day, I think, before they come back for the scans. I'm not very familiar with that, but I see a lot of deuterated H2O studies.
On a lighter note, try carrying D2O across customs and tell them there's a heavy water.
But for the, do you need a special receive coil for the deuterium?
Yes, you do. For deuterium, it seems like NMR is much easier than MR spectroscopy because you lock with deuterium. So we've been able to manipulate our NMR to do deuterium MR spectroscopy scans in cells as well as in vitro. But you need a deuterium channel, which also Siemens will help you if you're really interested in deuterium MR spectroscopy. But there's going to be a specialized RF coil, of course, which right now it's mostly in-house built, but I believe Rapid is also producing some of those coils.
Okay. So St. Jude lets you use it on volunteers?
Not yet.
Okay, yeah, I know the restrictions, so I was wondering.
We don't have a protocol yet, but the mice don't complain.
Yeah. So you have a mice-specific deuterium coil and everything in that case, right?
That's correct. We don't have a deuterium coil though. We use just QMRS.
Right. Okay.
But that you're doing at 7 Tesla?
It's no, we got 9.4.
9.4, okay.
Yeah. And it helps if you have a cryoprobe as well. It enhances SNR by a lot.
Mm-hmm.
It's a Bruker system, Puneet?
Yes, that's correct, Esin.
Okay.
So,
any other questions?
Yeah. Any other questions?
If I may ask, Esin, about your IDH mutant data, that was fascinating, actually. Are there any other metabolites than the usual 2-HG and lactate that you see are defining?
There are actually, when we looked at the metabolic profile, glutamate gets affected, GSH gets affected, but those are not reported as much as 2-hydroxyglutarate. There are some false positives and some false negatives of 2-hydroxyglutarate actually. They have been reported in the breast cancer models and also some, I think, brain tumor studies. So it is necessary to take a look at the whole spectrum, not only focus on 2-hydroxyglutarate. That was actually the idea behind using deep learning, for evaluating the whole spectrum instead of just trying to, take out 2-hydroxyglutarate and focus on that metabolite alone. When you look at the cycle, actually, a lot of the metabolites get affected.
Great.
Yeah.
Interesting. Very interesting. Thank you.
Thanks.
So I have one question. So last you showed like the peaks, right? Which you thought the model is picking up as important peaks. So did you use explainable AI for that?
Yes. So for the IDH mutation, we were able to identify choline, 2-hydroxyglutarate, lactate, creatine as important metabolites. When it comes to TERT mutation and TERT-only mutation, actually they were not as nicely defined. But I want to show them all, whatever we were able to gather. It is also not as nice for all the patients. This is an average of whatever we have, in terms of data. But actually, it shows nicely for some of the well-defined peaks.
Mm-hmm.
It is GradCam. So you can use GradCam for the spectroscopy dimension, just like the spectral dimension.
Okay, okay.
Mhm.
Another novice question. We're just talking about the time a patient spends in the magnet. Do we have something similar to time averaging? Do we have patient averaging? A cohort of patients that you average in a different distribution, right? You don't, a priori, you don't know which cohort they fall into, but you randomly pick groups, add them together, add the data together, and let the statistics guide to group them back. Is that something possible in this kind of, what's that?
Is it like a clustering type of application?
Yeah, clustering type, exactly, yeah.
Yeah, there is unsupervised methods. You could provide the information and then just ask the machine learning algorithms to cluster the data for you. But supervised learning actually has been shown to do better than unsupervised learning. But unsupervised learning or clustering techniques can be used to gain an initial insight into the data, what is available in it, and then go from there and possibly train models using that information. There have been several, techniques that actually applied different kinds of supervised and unsupervised algorithms back to back. So that might be an initial guess, or you can also use, unsupervised learning, because then it will be like looking at the data with no prior information and then just trying to understand what is in there.
Yeah.
It sounds like if this was a great discussion. And so I thank all our speakers. And if there's no more here, I'll declare the meeting closed.
Thank you for having us
Thank
here.
you, everyone. I enjoyed it very much.
Thank you for having us.